Hello, my name is Austin Bender, and I am an undergraduate researcher in the Ducker Lab in the Biochemistry department at the University of Utah. I have had the privilege to work with graduate student Kyle Dunlap and Professor Gregory Ducker on the research I'll be sharing with you today titled Intracellular Amino Acid Accumulation in Cell Lines Cultured in Amino Acid Deficient Growth Media. It is known that as cells turn cancerous, many metabolic processes are altered to support proliferation. Looking at data from the Cancer Genome Atlas, a databank of many healthy and cancerous human tissues shown in Figure 1a and 1b, we find that one such adaptation that occurs frequently in the development of melanoma is the downregulation of argininosuccinate synthetase 1, also known as ASS1, which is an enzyme that acts as the rate-limiting step in the conversion of citrulline to the amino acid arginine in the arginine-citrulline cycle of the cell as depicted in Figure 1c. When cells are unable to produce their own arginine, they become arginine oxotrophic, meaning that they become dependent on outside sources of arginine to continue proliferating. For this reason, methods to deplete a cell's outside sources of arginine have become attractive therapeutic options in the treatment of ASS1 deficient cancers, but have had limited results necessitating further study of arginine metabolism in ASS1 deficient cells. Looking for new ways to exploit potential vulnerabilities related to ASS1 downregulation, we examined the effects of arginine deprivation on naturally deficient and genetically engineered ASS1 deficient melanoma cell lines. During that process, we found that in low arginine conditions, ASS1 deficient melanoma cells displayed a phenotype that was characterized by an accumulation of intracellular amino acids when compared to cells grown in media that contained a relatively high amount of arginine, which we defined as a concentration similar to that found in healthy human plasma, as shown in Figure 2b. Interested as to whether this amino acid phenotype was unique to melanoma cell lines, we conducted a similar experiment that found that intracellular amino acids also accumulated in the LN229 glioblastoma cell line, which is also naturally deficient in ASS1 under low arginine conditions as shown in Figure 3a. Additionally, further experiments showed that the effect was not unique to arginine, but also occurred when cells were grown in media depleted of valine, another amino acid the cells cannot synthesize on their own as seen in Figure 3b. But the effect was not observed for cells grown in media depleted of glycine, an amino acid the cells can synthesize on their own as seen in Figure 3c. Those results suggested that the amino acid accumulation observed is a result of the depletion of essential amino acids, those amino acids which the cell cannot synthesize on its own, but not of amino acids the cell is capable of synthesizing, also known as non-essential amino acids. Curious as to whether the phenotype we observed was a result of a transcriptional or translational process, we conducted an experiment that took ASS1 deficient cells treated with rapamycin, an inhibitor of mTOR, which is a nutrient-sensing complex that when activated usually stimulates growth, and the translation-inhibiting drug cyclohexamide, and compared their effect on amino acid accumulation in the same cells grown in low arginine conditions as shown in Figure 2a. What we found was that cells grown in low arginine media showed a very similar amino acid phenotype to the cells treated with rapamycin, suggesting that the amino acid accumulation we've observed is related to translation in the cell. The accumulation of essential amino acids in the cell is speculated to be a result of an increase in expression of SLC7A5, a transporter that brings essential amino acids into the cell as a result of arginine depletion as shown in Figure 2d. Furthermore, an experiment which utilized isotope tracing, shown in Figure 3g, found increased synthesis of the non-essential amino acid serine in cells grown in arginine-depleted media, suggesting that an accumulation of serine is due to increased serine synthesis. With that said, we are still unsure of the specific mechanism and pathways behind the amino acid phenotype and many of the ways in which cellular metabolism is altered under low amino acid conditions. Moving forward, we look to explore pathways involving GCN2 and mTOR to determine their involvement, if any, in the amino acid phenotype observed, as well as additional isotope tracing experiments to determine how the cellular metabolism is altered under these conditions. We hope that by understanding the pathways by which ASS1 deficient cells respond to amino acid deficiencies, new vulnerabilities may be revealed that can be used in the treatment of cancer. Once again, my name is Austin Bender, and I would like to thank the Office of Undergraduate Research for their funding of this project, and Kyle Dunlap and Dr. Gregory Ducker for their support and guidance. And to you, thank you for listening.